So here we're going to be looking at the cytoskeleton and non-membranous structures within the cell. Kind of a cool picture here showing some of the interior structures that occur within a cell. So first off, we have ribosomes. So kind of an important component to cells in the sense that they are the site of protein synthesis within the cell. They're composed of rRNA, that's ribosomal RNA, and they have a large and small subunit, as we can see here, the large in the red and the small in the blue. And these subunits are made in the nucleolus of the cell. That tends to be the little dark circle you see within the nucleus. Now, one large and one small subunits make a functional ribosome. You need both components there to be functional to allow those proteins to be made. So if we kind of look at the image here, remember that ribosomes are three-dimensional, so that's why I kind of show this kind of three-dimensional shape here, and their important shape allows them to perform their very important function. So you have their messenger RNA that comes out of the nucleus, then there's two subunits of the ribosomes come together, and they allow that translation process to occur to allow messenger RNA to essentially be translated into a polypeptide chain, which ultimately will form a protein. Now, these ribosomes may be located either in the endoplasmic reticulum, thereby making it rough, as we can see here. These little purple circles here would be the rough. The rough would be the ribosomes and the rough ER. And they're also free in the cytoplasm. They can occur in small groups, or they can kind of be free floating. So they're not just attached to the endoplasmic reticulum, they can also be free floating. No matter where they're located, they're doing the same basic function. Now, ribosomes are RNA protein complexes composed of two subunits joined and attaching that messenger RNA in the center here. They're the site of that protein synthesis, and they're assembled in that nucleoli. And we see here the mRNA coming through, and our newly born protein, which is those linking of all those amino acids that are brought together by the transfer RNA here, and the sequence is very important for their function. Going into the cytoskeleton, in eukaryotic, cytoskeleton is a network of filaments and tubules that extend from the nucleus to the plasma membrane that support the cell shape and anchor organelles. It's kind of like that interior webbing to the cell. Helps it maintain its shape and assists its movement of the cell and organelles within it. It's assembled and disassembled as needed, depending on the particular functions that the cell needs to be carrying out. Now, within that cytoskeleton, there are three types of macromolecular fibers: actin, intermediate, and microtubules. So you'll see here, they both have kind of this very interesting hair-like kind of structural component view to them. Within those actin filaments, there's stress fibers that within the cytoskeleton, and they help with the cell move with motility. They're extremely fine, and they twist like pearl necklace here to help give them some strength. It's a dense web just under the plasma membrane that maintains the cell shape, and they support the microvilli and are also found in intestinal cells. And we also have with the cytoskeleton, as I said, in the small intestine. So this intercellular traffic control for moving components around within the cell, also cytoplasmic streaming. These kind of red lines represent the actin filaments, and their whole point is to kind of keep these villi apart. We don't want them all together. We want to maintain this high surface area to volume ratio, and actin filaments help that. Within those actin filaments, they can pinch mother cell into two after animal mitosis. So it's an important component in muscle cells also for contraction other than myosin. These are indications, these are very strong filaments. They're able to kind of pull those cells apart during the mitotic process, also important for your muscles when you're having contractions to occur, allowing that sliding filament theory to occur. Those have to be very strong to endure the stresses that those cells are under. We also have intermediate filaments, and they get their name because they're intermediate in size between actin and microtubules, and the rope-like assembly of fibrous polypeptides. Again, this twisting action can help increase their strength without necessarily causing a large increase in bulk. So they vary in nature, from tissue to tissue, from time to time. So these intermediate ones can kind of change a little bit. They do function to support the nuclear envelope, cell-to-cell junctions like those holding skin cells tightly together. So they do have some vital components, but they are not kind of one particularly set thing. They can vary a little bit from tissue to tissue and the time to time as well. Keratin, we might be familiar with the structure of our hair. It's composed of a lot of intermediate filaments. Nuclear plasma membrane connections, again, gives you that idea of how something can be very thin, but yet also very strong. And keratin is composed of those intermediate filaments, to give you a little bit there. And also, if you braid your hair, it can be stronger in that ways, and that's kind of what these can do on the molecular level. These microtubules, are intracellular transport, chromosome segregation, and these are like microtubules. They're like little tubes. They're hollow cylinders and made up of two globular proteins, alpha and beta tubulin. Kind of here's a cross section to kind of show you that kind of like tube that they do have in their appearance. Their assembly, they're under control of microtubules, the organization center. Most important is the centrosome when we're looking at kind of dividing a cell, as you see here. Interact with other proteins and cause the movement of organelles within a cell. So we want to just think about a cell dividing, and that's one, but also organelles need to be moving within a cell, and microtubules help with that. They're important for motor proteins. So what's a motor protein? Well, those help use energy from ATP hydrolysis to move proteins along the substrate, helping that movement. We talked about sliding filament theory. You're seeing that movement that's occurring, and microtubules are an important component for that. Those centrioles, there's hollow cylinders, as we can see here. They're short cylinders. They have a 9+0 pattern for microtubular triplets. It helps give them kind of like a collection of straws is a way to think about it, at least how they look visually. The centrioles found mostly in eukaryotic cells involved in the process of cell division. Centrioles may be involved with microtubule formation, disassembly during cell division, and in the organization of cilia and flagella, which I'm going to talk about in a little bit more detail. You can see, again, it looks almost like a circular form of uncooked pasta, for example. Now, these microtubule arrays of cilia and flagella, they contain specialized arrangements of microtubules. They're for locomotion. They help the cell move. Cilia and flagella share in common ultrastructure. However, cilia is only found in eukaryotes. That's an important note. Prokaryotes do not have cilia. So if you see cilia, you immediately know you're dealing with a eukaryote. Prokaryotes do not have cilia. And we see kind of the picture here of a flagella. Now, getting into a little bit more detail here, cilia, small, numerous. Flagella tend to be large and single. We see the flagella here on a sperm cell, and we have the cilia here kind of on the surface, look like little tiny hairs or appendages. Cilia and flagella both help move when the microtubule doublets side past one another. Each cilium or flagellum has a basal body at its base. It kind of acts as an anchoring point to help that cell move. The flagella are hair-like projections in the cell that aid in movement and are found in both prokaryotes and eukaryotes. Prokaryotic flagella are small, they're simple structure, rotary movement, and are proton-driven. Eukaryotic flagella, because both can have a flagella, they tend to be larger and more complex structure. They have a bending movement. So kind of think about it as one kind of like spins, like a rotary movement, and one is kind of just a back and forth movement. And that's to see the comparison between the prokaryotic and eukaryotic flagella. Now, in eukaryotes, cilia are much shorter than flagella. Cilia move in coordinated waves like oars, or kind of like the wheat in the wind, often used in preventing dust accumulation from your respiratory system. So that's kind of like act as a filtering component. And flagella move like a propeller or a corkscrew, kind of that kind of like rotary movement there, and commonly used for locomotion. Just as they would in a boat, they help the cells move as well. Cilia only found in eukaryotes for movement. You can see that forward stroke and that back stroke. You kind of have that back and forth kind of motion, like kind of kelp in the rising and falling tide or in the waves. For our eukaryotes, cilia and flagella motion, the flagella, as I said, moves in kind of a snake-like propelling cells, kind of back and forth motion, and the cilia have that direct back and forth. So when I say that motion of flagella, kind of like that propelling cells, kind of like that propeller, kind of spinning, if you will. The cilia have that true back and forth, that fixed point back and forth motion. It acts like a beating rate for about 40 to 60 strokes in a second, so they're moving very quickly. I have the picture here of kind of think of like the kelp underneath the ocean. It's a very slow movement, but the same general movement there. Lastly, the extracellular matrix. So it's secreted by cells. It's on the exterior portion of the cell. Large number of fibrous proteins. Collagen is one example. Might be familiar with hearing that in particular hair products. It's bound by integrins, so it's helping kind of like stick everything together, helping that cell communicate potentially with other cells and bind together. That extracellular matrix can also be important for communication between cells. So just another example of a component within our cells.